Hi Dr. G, it's Baven. I'm doing my project on the medical applications of DNA. An example of a medical application is recombinant DNA. Recombinant DNA is DNA that is synthesized non-naturally. One example that's very well known is insulin. The first step of making it is when the DNA is made from the protein sequence of the A and B insulin chains. The DNA polymerase makes a complementary copy of the DNA. Then the newly copied fragment enters the B-galactosidase gene on a plasmid with the tetracycline resistance. Galactosidase is an enzyme. It's a group of enzymes that catalyzes the breaking down of lactose into its monosaccharides. As a tetracycline is an antibiotic that stops protein synthesis. Tetracycline is then added to get rid of any part of the plasmids that have not turned into the bacteria. Now, the normal bacteria would normally secrete or let out the B-galactosidase into our body, and these new bacteria are grown, and then the bond between the galactosidase and the insulin within the plasmid, that section is harvested. So now that we have this chain of protein, the galactosidase part is taken off. In the right conditions, when these two proteins mix, the bonds between them form usable synthetic human insulin. Another very famous medical application of DNA is DNA microarrays. Microarray is a way for scientists and doctors to see thousands and thousands of genes on one microscope slide. This has led to earlier and easier detection of cancer and strides with heart disease. Citable by Nature Edition says it defines DNA microarray technology as saying that they exploit the ability of complementary strands of nucleic acids to base pair with each other and bind. It gives an example, and then it says that this method was first used by Saul Spiegelman to measure the homology of two different nucleic acids. Spiegelman called the method hybridization of nucleic acids. Later, the developers of the DNA microarray dotted an array of DNA copies corresponding to a large number of different mRNAs of known sequence onto a glass slide. Because this array was so tiny, it was termed a microarray. Although the cDNAs were double-stranded, they could be melted or denatured to single strands, which could then be used to bind or hybridize to fluorescently labeled nucleic acid samples from cancerous or normal cells. After washing away the unbound molecules, bound fluorescent nucleic acid samples were identified by laser microscopy. Fluorescent dots indicated expressed genes, and differences in microarray patterns between normal and cancer cells could be quickly identified. And within the large spectrum across which DNA can be incorporated into the Human Genome Project and medical applications, I have focused mine on Progeria. Progeria's name comes from the Greek roots of pro and geras, meaning of premature old age. That's when that occurs. And when the lamina is mutated, it kind of destabilizes the membrane and causes it makes it very, very susceptible to die prematurely.
We have cloned the Progeria gene already. It's in the human genome database somewhere. We have to find a way of pulling out that Progeria gene from the mass of data. It's a lot like hunting a serial killer. You know the killer is out there, but you don't know what he looks like.
Some of the symptoms of this are baldness, very swollen veins on the head and arms and legs. They tend to have no eyebrows or eyelashes, a very beak-shaped nose, a small shrunken chin, premature cardiac disease, very narrow, usually ribs are showing in the chest, and very swollen and stiff joints and age spots. This is an excerpt from a speech given by Dr. Leslie Gordon. She has been the main drive behind the Progeria Research Foundation, and She is the doctor who kind of started all of this. Around the late 90s, she began, and then by 2003, she was the doctor that led the discovery of the gene and therefore led to the official DNA test for progeria.
Now, as most of us here know, Hutchinson-Gilford is caused by a single base substitution, which leads to the use of a pointer. Okay, which leads to the use of a cryptic splice site and cuts out 150 bases from pre-laminate, the laminate transcript. So here's the normal splicing, and here's the abnormal splicing, and this is missing. The result is an aberrant pre-laminate protein, which we refer to as progerin, and it's 50 amino acids short. And there's a lot more of that biology that goes along with it, but I think that's going to be covered over the next couple of days. But that loses its recognition site for the ZIMST enzyme and becomes permanently farnesylated, and hence permanently intercalated within the internuclear membrane. And all of these things were discovered almost within the same time period in the same couple of papers.
And the next step they're working on is the cure.